this is not part of the lecture. Yesterday I was showing you two different types of filter, GFF and polycarbonate. So this is an image of a beautiful coccolithophore. But what I want you to look at is these pores. So this is a polycarbonate filter. With the polycarbonate filter, there's the little white one that I pulled out for some of the groups and show, and I can show it again. And these little dots here are the pores. And I cannot remember what did I filter it on, 0.2 or 0.45. But I mean, you have a scale here, so you can see the filter. So this is how the polycarbonate filter looks like. This is how GFF filter looks like. This is a different magnification. So it's 10 times. So this is 2 microns here. This is 20 microns here. But this is the structure of GFF filter. So just so you guys know, when we were talking about, specific pore size or like average pore size, because we know how much GFF, how much glass fiber filters that we put on, and that's how it's kind of measured to have a certain pore size. So just so you know that these are two different things that you deal with. Okay, optics. Let's go up. So today I'll talk to you about different type of optical proxies for the IOPs, actually for in situ measurements. I'm not going to go so much into AOPs because that's what you guys are going to be covering next week, and then we're going to be talking about remote sensing, how to derive these proxies. But I'll just kind of like start building on the idea and take you somewhere. So if you imagine any experiment that you may have done in your life, or a cruise, or just think about an experiment that you want to set up, you're always going to have a certain number of parameters that you want to measure. And not all the parameters are going to be able to measure on the same frequency, the same number. There's always going to be lots of parameters that you can measure really low frequency. So you can collect just maybe 1 or 2 samples per experiment. But there's going to be a really small number of parameters that you can measure in high frequency. So you go to see, you do a profile with the CTD, you're going to have temperature, salinity, and high frequency, lots of numbers. But when it comes to discrete samples of chlorophyll or discrete samples of HPLC, either because of the time consuming or the price of the sample preparation and analysis, you're going to collect a lower number of those samples. As it comes out to be, it's always biogeochemical samples are the ones that you really want the most, in my case at least, but there's only so many that I can collect. So what you really want to do in order to jump around this stuff is to make this connection between the physical measurement and biogeochemical measurement. And that's what we're kind of trying to do in this class, to establish these relationships between the physical measurements of the light or behavior of the light or behavior of the particles when light is shone on them, and the biogeochemical parameters of the interest. So this connection allows us to artificially, through a relationship, increase the number of the samples, increase the frequency of the samples of the biogeochemical parameters that you're looking for. And that's what proxy is. And an official definition of proxy is one thing, but when we talk about proxy in optics, it's a measurement of physical quantity optics that is used as an indicator of the value of another, in this case biogeochemical property. So that's what we're going to be talking about today. Now, different ways that people came out to use optics to study different properties of the biogeochemical processes in the ocean. There's many papers out there, tons of papers out there, and I just did a selection of them. And I made a special homage and tried to select only, well, not only, but predominantly the papers that were done by the graduates of this class. So you guys know what is expected of you when you come up here, when you come out of here. So yeah, the title, it's Carbon and Other Biogeochemical Proxies. I'm going to be using lots of acronyms and introduce you to them as I go. I just note this satellite image. Doesn't it look like some kind of scary monster with the eyes coming out of it? So it's Alaska up there, and really beautiful swirls and twirls that you can see. So why do we want to develop these proxies? Using optics, using these physical measurements, we get much higher resolution data set, And traditional methods are always of the sampling of the biogeochemistry are often expensive and time consuming. You guys know how long did it took us to filter yesterday. That 900 milliliter sample, the group that was there, I think probably still filtering. I think it took us like a good 4 hours to filter it. So it's time consuming. And then if you want to take that sample and process it, for example, HPLC, it costs you $100 a pop. So it really doesn't allow us to establish some knowledge about the biogeochemical processes that we're interested in. And if you look at this really, really cool graph that was originally published by Chang and Dickey, and you're going to see many different, maybe there was somebody else, but this is where I found it first. You're going to see many modifications of this graph. It actually plots different processes in the ocean on this relationship between the spatial scales, horizontal spatial scales, and temporal spatial scales. And if you start looking at the processes that we've been talking about a lot, so anything that has to do with the phytoplankton and things like that, they're going to sit in this gold center in the spatial scales from 10 meters to 10 kilometers and 1 day to 1 month. So if we would put a physics lingo here, there would be sub-mesoscale, mesoscale processes. So think about it if you have a capability of going, out with the ship only like once a week, there's a big chance that you can actually miss a bloom. So if you have a mooring there, this mooring is going to continuously measure the chlorophyll concentration, so you're not going to miss a bloom. So this is the idea. You use optics to artificially increase the frequency of your samples to sample the parameters on the scales of importance. Because if you don't see something happening, doesn't mean it's not there, no? There's a story that I used to use with my students about a thing that happened in my house. When I used to live in LA, there was a cat, and we would give it food, and the cat would come and eat the food, And the cat didn't eat too much food, and suddenly the food started disappearing all the time, And I'm like, I feed the cat, wake up in the morning, cat's looking in the empty bowl, surprised as I am, So there's a couple of hypotheses that built. I'm like, cat is bringing the buddies because it's inside an outside kitty, but she wasn't a friendly cat, Or she's just eating a lot, she has an internal parasite, What it turned out to be that I had 2 possums living in the house who were growing really nicely because they were fed by high protein food. But I had 2 hypotheses that I came out to be, which were most plausible based on my previous knowledge of behavior of the cats, But it was something completely unexpected that was happening. So if I had a camera there, a constant presence that was feeding me data, I could have known. But I could have published a paper on, fantastic social skills of my cat, to sneak in buddies and take them out. So that's the importance. So really, if you want to study the process, think about the scales on which do you have to study and proper tools. So, and one other really important reason is that when you think about remote sensing and models, we have remote sensing, we want to validate it. And sometimes it's impossible for us to collect so many discrete samples. So by developing this really strong relationship between the discrete samples and IOPs, these in situ optical properties that you can measure in the water, you can use them then to validate remote sensing high-resolution biogeochemical models. And Niels just has finished his thesis and about to publish a paper where he did exactly that. He developed, he used the pre-existing idea and used some samples to develop a relationship between the data that he collected on BioArc afloat inside an ocean in order to validate and check for the remote sensing algorithms, how do they perform in that area. So first highlight of the graduate. Further down, why are we going there? So these optical instruments are getting smaller and more robust and cheaper, and energy cheaper. So we can deploy them on different platforms over extended periods of time and in hard-to-reach areas. And I think this is the second time I'm giving this talk, and every time I'm giving this talk, I'm adding more and more platforms because there's more sexy things coming along. And again, it's not only the choice of the instruments that you will have to make, but also the choice of the platform. The platform on top of which instrument will allow you that focus on the sub-mesoscale, focus on the vertical, focus on the intensity of the sampling that you really want to do. So for example, this is a drawing of the wire walker, which is kind of a cool Lagrangian Kano-style buoy that has this oscillating package on it, and it can do, I don't know, like a profile up to 150 meters in 7 minutes, and does that continuously for only 3 days. So think about your experiment, that stuff. Or you can have a glider that can be deployed up to 3 or 6 months, depending on the type, but it glides a little bit slower. You can go in Tara and set up this flow-through system and collect the data all over the ocean, so on. Sail drone, you can have your buoy, your float, so on, so on. But again, you can dump them in there, they can ride out the storm, they can ride out the winter in North Atlantic and actually give you some meaningful data, if they come back, and if there is a connection. How do you do that? This is something that Colin said in 2015, and I think I find it really, really inspiring. I think she said it again today. I think she always says it before I have to give a talk. is that anything that causes variability in the sample is an opportunity to extract additional information from that sample. So that's what you just said this morning. Okay, you guys think that the whole thing with bleach is messed up. Well, try to think about it a different way. Try to use this like potential disaster into opportunity. What is the other information that gives you? Because if you only think about chlorophyll fluorescence versus HPLC, I mean, it's going to get you good validation data, but it's not going to get you too far. Try to think outside of the box. That's what you guys have been, think within this constraint, but outside of the box. So proxies in general can be, icon in my head, divide them in two groups or several groups. First one, where you try to define like amounts. How do you call it? English word seems to disappear for me. Bulk properties, like concentrations of things. So we can talk about in that point about quantity and quality, So for the first order chlorophyll, we all know we can use chlorophyll fluorescence, we can use absorption, as you guys seen that. Particulate organic carbon, we're going to talk a little bit about that later in the lab. Particulate organic carbon, although it's a small portion of the surface pool, or just generally pool of the carbon in the ocean, is extremely important because it contains phytoplankton. And that's that magical connection between that inorganic carbon that is transitioning into the organic carbon then feeding the ocean. So we really want to know, have a way to measure it. Phytoplankton carbon also can be derived using different IOPs. Suspended particulate matter, total amount of mass of the particulate that you're encountering. It's really important for the estuarine and close coastal shelves and just turbid environment. And it can be really nicely correlated, as Emmanuel was showing you earlier, with CP and BBP. Particularly in inorganic carbon, important for the carbonate chemistry, coccolithophores and so on. There's a beautiful method from SI-labile backscattering that Barney Boucher is going to come and give you a talk about it in week 4. He does that really, really successfully. You can use cross-polarized attenuation. Emmanuel mentioned it. You asked about it, I think. Is that the thing that Bishop does? But he's actually taking the attenuation meter and puts a polarizer. And just because of the way that minerals of the calcium carbonate polarize the light, you can actually use that opportunity to quantify in situ the amount of calcium carbonate. You can look at dissolved organic carbon through the COOM, but as you're going to see, it only works in certain areas. You can even go to the chemistry, and that's something that Colin mentioned about the absorption of the things in UV region. It's not just the organics that absorb. And you can go all the way to the primary productivity. Use some idea about the variable fluorescence, some DL variability. Then you can start talking about the quality of your things in the water, about the composition. You can look at different ratios of the scatterings and backscatterings. And Emmanuel spoke about that, yes, and get an idea about the composition of your particles. You can start doing some, distinction between mineral particles, phytoplankton, even different phytoplankton types. You can try to derive particle size using slopes. Emmanuel spoke about it yesterday. But some kind of also crazy ways of looking at the fluctuation of the signal over the mean and try to connect back that to the size of the particles. You can play with the multiple angle CPs, multiple angle scattering, and start looking at the spectral slopes and in the spectral slopes, this VSF pretty much, and start saying something about it. I mean, there's so much more in polarization. We would go there. We can also start talking about some aspects of the quality of the particles if we go in that direction, but I will not go in that direction because I'm not capable of going there yet. I will learn. Phytoplankton composition, going back to PFTs, you can play with different ratios of the chlorophyll to carbon, multiple channel fluorescence, because then we know that they have different pigments. They will fluoresce differently. Go with chlorophyll, look at distribution of the chlorophyll, look at the APs, and so on. And then DOC type. Different type of dissolved organic carbon will have, if they're colored, different fluorescence signals and different slopes. So I'm going to touch upon some of these. But then this is the exciting part that I really like a lot. Not only that you can define these quantities now, but you can start defining the fluxes between these quantities, the fluxes between the pools. Once you're secure with your measurement of these bulk properties, you can start looking at the changes. And that's pretty cool. That's kind of what you're interested in these biogeochemical studies. You can start looking at these movements of the quantity from one pool to another. So through space, so just carbon export from the mixed layer to the deeper ocean through time, which gives you the idea, like let's say productivity, such as primary production or different types of productivities, or type. The switch from different types of carbon pools, so from the phytoplankton to the trial matter, or from the particular organic carbon to dissolved carbon. So kind of this is where you want to go. I'm talking too fast again. Everybody yells, I slow down. So I think somebody said once, the chlorophyll is not a chlorophyll is not a chlorophyll. Doesn't make sense. But as how many every times they're going to hear us saying chlorophyll in acetone versus chlorophyll in methanol versus chlorophyll in cell versus happy chlorophyll in cell versus sad chlorophyll in cell is a completely different story. And all these different aspects that are chemical, physiological, or compositional will be seen in these spectral parameters that we're trying to measure. So just mind that. So this is a paper by Colin and Andrew Bernard, where they looked at the line height that we see in absorption of the phytoplankton and actually built a really nice relationship between the A676 and chlorophyll. So if you have this stable relationship, as they showed, it works in Gulf of Maine, in some cultures, and so on, you can try to apply similar type of relationship to your ACS data. So what Emmanuel's student, Ali, who's also graduated this class, and she's going to be with you next week, I think, when she comes back from partying. She's on a wedding. She took this relationship and applied it to ACS. So she compared the number of samples, just for this graph, where she has chlorophyll derived from HPLC. She has 402 samples distributed throughout the globe. And this is really beautiful HPLC data. So this is really big. When she used this relationship, she increased the number of her chlorophyll samples to 96,000. Actually, that's 97,000, no? So several orders of magnitude. And it's a pretty stable relationship. She has characterized uncertainties and stuff. And I mean, if you look at this distribution of your chlorophyll as a function, frequency of your chlorophyll as a function of concentration, it fits really nicely. That she can do cool stuff. And now when you have this data set in hand, you can go wherever. And she really went a nice direction, and look out for her paper. It's coming out soon, hopefully. You can look at the chlorophyll biomass using fluorescence. Colin again. Published just recently a beautiful review paper where she, with her colleagues, including Emmanuel, a bunch of people, looked at a bunch of different papers, a bunch of different data sets from all over the world, as you can see here, and they looked at the chlorophyll, fluorescence, versus chlorophyll derived from HPLC. And here it's plotted in log, so it looks really nice. Here it's plotted in linear and looks not that nice. And it's, once again, think about it, what we said. Once chlorophyll is in the cell, it depends on the physiological status of the cell, happiness of the cell, compositional things of the cell. And that's what they went and investigated further on. For example, if you look at the physiology of the cell, so light, nutrients, different life stages, you can plot this chlorophyll fluorescence over chlorophyll as a function of light. And what you're going to start seeing is that non-photochemical quenching that you're going to learn about next week, where the cell is kind of pretty much freaked out and start turning off the capability of capturing chlorophyll. And you have those pigments that are protecting the chlorophyll from them and jumping in. And they can be seen as a drop in the fluorescence on high levels of light. Species composition. So what here on the y-axis, what you have is a slope factor. So they're looking at the calibration from the factory versus calibration from the lab. So just comparing that stuff. And they're looking at the variability of that slope as a function of different types of phytoplankton, now grouped based on the lineage of origin of their plastids. So if you guys remember when I was telling you about pigments, I was showing you this like evolution of the plastids. You had cyanobacteria there, and evolution of plastids in red lineage and green lineage. And you see some differences, no? So think about it. And then in order to actually get meaningful chlorophyll concentration out of chlorophyll fluorescence, I once did it for one paper, and there was lots of massaging that I did. When I plotted that chlorophyll fluorescence versus chlorophyll, it looked really bad, you know? But then we have done fourth level correction. So we corrected for the PAR, so removed non-photochemical quenching. Then we looked at the temperature. And temperature in this situation was a function of nutrient availability. And we corrected for that. And then we corrected for depth, again, nutrient availability, and then time, because over time we had a change in the species composition. And they gave us a really kind of nice fit between chlorophyll that we measured and chlorophyll derived. However, we just used that to interpolate the data. So we're really not comfortable extrapolating, because I know for a fact that there's a species composition that's going to drive that change. So if I start extrapolating it to the area with new type of phytoplankton compositions are going to come, a new nutrient limitation is going to pop up. I'm not sure if this is going to hold anymore. So that's kind of know your strengths and things like that. There's many other papers that I haven't highlighted here. For example, Jing et al., they used radiance, just downwelling radiance, spectral radiance to collect, spectral, to correct that chlorophyll fluorescence that they had deployed on a float. And you can see this reference and kind of Google it a bit. I'm going to put all these papers in your Dropbox so you can just start getting ideas about different ways how you can massage the chlorophyll out. But just remember, you know, kind of interpolate, don't extrapolate.
So where is this example from?
This one? That's from find my paper, that's 2015. Sorry, I didn't put the reference. It said,
What region?
that's North Atlantic. This is from North Atlantic bloom. This is everywhere, North Atlantic bloom. This is everywhere. So the data that I'm showing for North Atlantic bloom are also included in this graph here.
And that's like the spring bloom?
That's spring bloom. So there was a transition between the diatoms to some kind of pico-eukaryotic composition. During that period of time, you had lots of nutrients, you had some deep mixing, then you get a shallowing, silica ran out, the little guys start coming. So that's all captured in this graph.
So you're worried that a change in season or place might damage?
Well, yeah, completely. I mean, again, chlorophyll is not floating in the ocean. It's embedded inside of the protein, inside of the complex, inside of the membrane, inside of the plastid, inside of the cell. And all these things can impact on that poor little chlorophyll and how it fluoresce. You know, remember the ratio of how much light goes into the heat, how much light goes into photosynthesis, and how much light does it go in fluorescence is highly dependent on the physiological status of the cell.
So has anyone checked these like in lab, controlled lab experiments as opposed
Oh, yeah.
to temperature?
Oh, yeah.
Then we could sort of
But do you think that you can apply that stuff? I mean, you really in the oceanic environment, you have just one condition that is driving the thing. There's so many conditions driving. So I have never seen, you know, it's really hard to do experiment that you can, you know, try to tease out like 5 or 6 different drivers. There's many different lab experiments I can put some things on, and you can easily do them. Colin did a couple of papers where she actually looked at certain conditions, and some earlier papers where they looked at that stuff. Yeah, but again, the response to the temperature of your Hitoceras culture, we're going to look at the fluorescence versus chlorophyll. It's going to be completely different than the Dunaliella culture. And then if you put Dunaliella culture in silica limitation, they don't care because they don't need silica. But on the other side, Hitoceras needs silica, so it will react. Then if it's the iron limitation, it's going to look differently than the silica limitation. If it's a nitrate limitation.
So I'm getting a little bit scared here.
Don't.
You know, global mass and
Yeah, well, good, you're thinking. Be scared. No, be scared, be careful. You know the constraints. That's what I'm saying, interpolate, don't extrapolate. I mean, if you're saying and there's a coccolithophore bloom offshore California, I can tell you for a fact that it's not there. It's not there. So, you know, like before you start applying and taking somebody else's algorithm, make sure you understand what it's doing and test it in a couple of places and see if it doesn't make sense. If it does, great. If it doesn't, you know, just don't. But don't be scared. And just be critical. Be critical. Be, you know, pragmatic. That's you know, that's what you're a scientist. You're here to learn how to be critical of everything. Don't trust a single thing that I'm telling you. I mean, I might be just here by accident, no? So, and primarily don't trust yourself. I don't trust myself. So this is kind of chlorophyll. So I'm going to just switch slowly to carbon. And I'm just going to do like a little highlight of the cool things that I like, you know? And carbon is something that I do a lot. So Gardner, Wolf Gardner, who's the king of carbon, particularly organic carbon, and its relationship with attenuation. He's the first one who actually did this measurement. This was done on original JGOFs in North Atlantic, North Atlantic bloom experiment. He first time measured attenuation versus particularly organic carbon. The idea of this relationship is that all the particles scatter. In the oceanic environment, most of the particles are carbon associated. Therefore, you should be able to make a meaningful relationship through which you can correlate these two. So he's the first one who did it. And since then, a bunch of people did it, including me. But he will come to the lobster bake, and you want to talk with him about lots of stuff. I uploaded some papers. I will upload more papers from Gardner. He did some beautiful review. He's a walking body of knowledge. Very nice guy too. So take your time to talk to him. So the idea is here that we know that both attenuation and scattering, and backscattering, are associated with the particle concentration and size. However, backscattering is also highly dependent on morphology and type of the particle. Carbon density in all the particles that we encounter in the ocean is not the same. So if you say, well, then I'm not going to go backscattering because I know that attenuation is much more connected to the cross-sectional area, I'll go to attenuation. But the carbon density in all the cells is not the same. You don't have the same amount of carbon diatom in your Hydroceras per cell and Dunaliella per cell, or per biovolume per radius, just because they're two different creatures, no? And you're not going to have the same amount of carbon detrital thing versus phytoplankton. So there's variability. Just you have to think about the uncertainties of your thing. So what I did in one paper, that I looked at the carbon, particularly organic carbon versus backscatter, and particularly organic carbon versus attenuation here. And the dots that you have here in my samples, the red lines are my relationships, and the other lines are the previously published things. And you can see how, beam C looks nicer, nice tight, everybody's happy in this region, everybody's tight. But when it comes to backscattering, it goes all over the place. And that goes back to the point that backscattering is so much more dependent on the morphology of the cell and different internal structure and external structure and mineral type of the particles. Well, beam attenuation is kind of really looking at the cross-sectional area. So when you start decomposing this beam attenuation, you actually start getting to see some trends. So what I've seen is that this relationship, the slope of this relationship, is highly dependent on the type of the particles that I'm encountering when it comes to the open ocean types. If I'm seeing larger particles, this slope of this relationship is going to be lower than the one with the smaller particles. If I'm in oligotrophic systems, and switching to eutrophic system, that's going to change again. And so I saw this relationship and start populating with the different publications previously, and except for some outliers that are associated with the methodology, everybody's following the same route. The scatter that you see in this relationship can be attributed to the natural variability or to the difference in the methodology. And sometimes those things are really hard to discern. You really have to, once again, understand your method, understand everybody else's method, and understand, as Colin and Emmanuel were saying, how much of each of these steps, how much uncertainty each of these steps in this processing, collection either of the particular organic carbon or your optics is bringing. You know that different type of filter size, so GFF versus polycarbon, is going to bring 3% error. And when you start understanding that and start quantifying that error, then you can start eliminating the scatter and kind of trying to tease out natural variability. And so what this is saying here, just because of the density of the carbon in different particles, is that there's a trend in relationships. So when we see a shift towards diatom-dominated types of water, this relationship is going to be, the slope is going to be lower than for the pico-eukaryotic type of waters. Why? Now we're going to go back to the biology. Diatoms are different than anybody else. I'm just going to cover this really fast. When you look at the diatom cell, there's like so much optics here, there's no space for biology. If this is a diatom cell, the majority of the cell is a vacuole. It's a gigantic vacuole inside. And there's teeny tiny, all the plastids, DNA, everything. It's kind of like squished. So the majority of the cell is actually vacuole. What that means that if I compare the amount of cellular material per cell in my diatoms and my Hydroceras versus my Dunaliella, I'm actually going to get higher carbon density in Dunaliella versus the diatom, regardless of the cross-sectional area. So what you're seeing here is that the transition towards the diatoms, you are, for the same cross-sectional area, increasing the amount of carbon. And it's kind of hard to understand, so I'll direct you to the paper. But just what you're seeing here is actually driven by the carbon density of the particles. you can look at the relationship between suspended particulate matter and CP and BVP. These are also particles, they scatter. And there was a great paper by Greet Nuckermans, and it's from 2012. Well, she looked in a very similar way like I did, the relationship between the CP, BVP, so attenuation and backscattering, and suspended particulate matter. And what she found that in these two cases, in case of CP, the relationship is highly dependent on the particle apparent density. And for the backscattering to suspended particulate matter is on particle composition. And it's kind of similar to what I've seen to the carbon. Now you're really in a particle apparent density, carbon density, here particle composition, the same thing for the backscattering. I didn't really cover that too much. And this is the graph that you guys saw yesterday, and Emmanuel has shown you that attenuation, the slope, the relationship between attenuation and suspended particulate matter as a functional increase in SPM is following this direction. So it kind of looks similar to that plot that I've made, no? So you can start comparing them, and you can see that these things are actually flipped, no? So where are we seeing the opposing trends? So here I'm looking at the particular organic carbon versus attenuation as a function of increase in mass, and here I'm looking at the opposite slope as a function of increase in mass. You guys might know what the difference is. Let's see the first order. Let's look at the numbers that they're looking at here. This is gram per meter cube, no? It's going to make a point, but these are much higher concentrations. I'm here sitting in the open ocean. If this is gram per meter cube, that means here you have a kilo, 2 pounds of suspended particulate matter per meter cube of water. That's a lot, no? And here I'm really working really low environment, low oceanic environment. So what you have here is a biology driving this relationship, and here is a mixture of the optics and actual like high-end loading of the particles. We can go through this a little bit when we're going to be filtering SPM and POC, and I can go through this in details. But it's really cool because now you can start, looking at this relationship not only to understand how you can, take this now CP and measure carbon everywhere in your floats, in your vertical profile, and so on. But you can also start looking at that slope as a function of different phytoplankton types or different amount of particles that you encounter in a system. Phytoplankton carbon is actually the thing that we really want to care the most, because at least from my perspective, no, because that's where I'm going to take off my shoes because they're wet, terribly wet. So, because this is what you want to put inside of your primary productivity model, and this is where you want to. This is where everything starts, where you can feed your export model, you can fit in your climate models and your Darwin models or whatever. So there was a really cool paper by Jason Graff and colleagues, who's not a graduate of this class, but I'm sure, but Emmanuel is on that paper. He's not. Toby is on that, but Toby's not a graduate. Toby? Giorgio. George is a graduate class. He was on this paper. Okay, I found one. It's like I was really trying hard. And if we look at the global relationship between the backscattering and phytoplankton carbon, the way he makes phytoplankton carbon measurement is really strenuous and long relationship. He's using this flow cytometer, this really specialized flow cytometer that is by looking at the fluorescence peaks and the size scatter. There's a measurement of the size, differentiating different particles, and puts them in. Separates them in little tubes, and then he collects a bunch of these little cells in a bunch of little tubes, and then he runs that for the carbon. And he knows exactly how many cells is there in a carbon, so you can make a really good measurement of that carbon. What you're doing with POC, you're measuring everything. Particularly organic carbon, we're going to capturing all the particles in a filter, so it's going to be detritus, bacteria, whatever came along, zooplankton, whale, whatever. Here, he's actually taking the cells and measuring actual carbon in a cell. So it's a really highly precise measurement. However, it takes some time, so this is an amazing amount of data points he has. And what he's seeing, and there's a really nice relationship between phytoplankton carbon and backscattering. And then you can take this and apply it again to your floats, apply it to your I don't know, remote sensing, whatever, and see this phytoplankton carbon everywhere in the ocean. There's different ways to get to this phytoplankton carbon. Doesn't have to be this precise. Most of us don't have the influxing capability, so we can use different methods, including taking the images of the phytoplankton, calculating biovolume, and then deriving carbon from there using formulas, and then actually try to make similar relationship. But this is a true analytical relationship. Again, so now we're going from the realm of the particulate into the dissolved. We all like sediment, but really, if you're a carbon person like me and you're studying geochemistry, biogeochemistry, you really want to know dissolved organic carbon. And, this is a big level of discussion, and without knowing what is in your water, you can't say a lot, but many papers have shown that sediment, to a certain extent, is in some areas of the coastal ocean really replicating dissolved organic carbon. And if you think about the sources of dissolved organic carbon in the coastal ocean, concentrations that you have it, you should be able to make some kind of meaningful relationship between the absorption of sediment and concentrations of the DOC. And that's for all of you who are working in like estuarine systems or swampy areas. This is something that you might encounter, no? But when you come to the open ocean, this thing starts falling apart. First, the concentration of the sediment, in open ocean is really low. The type of chromophoric, just the type of color, the organic matter is just really low. Just generally, DOC is low. So there's just really not spectral characteristics in that sediment that is relatable to the total DOC concentration. And this is a paper by Nelson and Siegel, and I think they always send students over, let's put it that way. They never were graduates, but there's always one or two students here sitting. There's one there. And they've done this really beautiful work where they collected tons of these samples that has really shown that throughout the world's ocean, you can't make a meaningful relationship. And there's many reasons for that. One of them is composition, but there's some other ones that you might discuss next week. Okay, so now we can go talk about composition. So how, different cool ways that you can derive some knowledge about the quality of the particles from using IOP. I can remember Emmanuel has shown this graph yesterday. Yeah, you have yesterday, no? And this is a cool paper by Twardowski, who's not a graduate, but he calibrates our he calibrates our instruments. You're in this paper. You are in this paper, Emmanuel. Okay, cool. Now I'm just trying to make a connection, continuous connection. And Emmanuel explained this yesterday. You can start combining different optical properties in order to start discerning different types of particles that you're encountering in your system. So here, by combining, backscattering ratio and Young slope, and plotting the index of refraction or whatever that is. I think that's an index of refraction. Yeah. He started differentiating different type of particles that he encountered because they have different type of spectral characteristics. They're visible in backscattering ratio, and they have a different size range. They're visible here. So this is kind of pretty cool. Something that you could try to do while you're doing optical lab is collecting data. Why you Yeah, exactly. So we started looking at fluctuation of the signal around the mean as a function size of the particle. And we did it from atmospheric particles. So Wayne and Nathan started looking at trying to use this stuff to check it out. Yeah, and then he discovered, yeah. But Nathan does research with the daily paper. And it actually takes him one time to publish, to start reading papers, and thinking goes fast, and that's what he does. So without reading the paper, he developed the same thing with Gutenberg, but it's the same idea. With the variability of the signal around the mean, eventually determines on the hydrogen level.